The next speaker is Rita Dias, Synergism of DNA-binding agents and macromolecular crowding on DNA condensation.
Good afternoon, everybody. Yeah. So I also come from Norway, and well, I have to say that the temperature indoors is a bit low for me here. So I got a sore throat. So one of my goals today is to actually keep my voice throughout 30 minutes. But anyway, so this is the title of today's talk. And I'll just start by, again, on the principle that I might not be able to reach the end. I'll just start by sort of acknowledging the people that have been working on this project. And this work has been mainly developed by Shravani, which has been my PhD, or one of my PhD students that finished last year. And then we have Ramilala here, which works at the biotech department in NTNU that helped us with the DNA and the protein, and some collaborators in Brno University of Technology.
Sorry.
Yeah.
I'd like to pin it here because your head is over.
I'm sorry. Right, so let's see if there's this better. Yep. Okay, so we've seen quite a lot of interesting talks yesterday regarding the condensation of DNA in eukaryotic cells. So we've seen all this wrapping of DNA around nucleosomes. And what we're mostly interested is on the DNA condensation in bacterial cells. So, and this looked very different. So if we just look at this very simple representations here, one of the main differences is that we don't have the cell nucleus is not present, so there's part of the confinement is gone. And instead of having this very particular histone proteins, what one has instead is a combination of proteins. And in addition to that, so this is and there's a few different parameters that are believed to be important in DNA condensation in bacterial cells. So we have the DNA supercoiling, which is sort of represented here. So there's a class of enzymes that are responsible for coiling up the DNA inside bacteria, twisting it and so forth. And then we have, then what I was mentioning, the architectural proteins. So they're called architectural proteins because they have different binding modes, and they introduce different changes to the DNA. So you can have bending or wrapping or bridging. And they're often also called nucleoid-associated proteins. And apart from these two, there's also the molecular crowding effects. So again, we don't have the nucleus around the genome in bacteria. But however, we have quite a large concentration of proteins, plasmids, ribosomes, which are present in the cytosol, and that are believed to be important in inducing or helping in this condensation. And this was initially shown by Zimmerman and Murphy already in the mid-90s. And this was shown, for example, by the fact that, yeah, again, we have a very large concentration of macromolecules in the cytosol. And, for example, that cell lysis is accompanied by DNA unpacking. And there's been also quite many reviews on the topic, more reviews than actual work, to be honest. But yeah. So what we've been focusing on, and also I should also mention some recent work by Joyot doing simulations where suggests that there's some segregative phase separation between the highly charged DNA and the highly charged ribosome. So that's yet another mechanism. So, and in the spirit that Anatoly was telling us yesterday, so we've decided to focus on two of these aspects. And the two aspects are the protein, or let's call it a DNA-binding agent, that we're going to change, and also then on the crowding. So we're focused on these 2 points. So Shravani, she is very, very interested in molecular biology. So she really wanted to work with a protein, and she sort of chose to work with H and S. And there's a lot of work conducted regarding the interaction between DNA and H and S and the different binding modes. I'm not going to go into details. We might hear about that tomorrow There's some work done here also in NTU on that. But I just want to sort of very briefly, because we don't really focus so much on the binding mode, but so HNS comes from histone-like nucleoid structuring protein. It's a relatively small protein, 137 residues, and it's found in relatively large concentrations, or is one of the, depending a little bit on the cells and the cell cycle, but it can be found in reasonable concentrations, and is believed to have many different functions in the cell, one of which the regulation of gene expression. And when it's overexpressed, the genome can get too condensed. And, the protein itself has one part that is that has some specificity to DNA, low to medium specificity to DNA. So it has a DNA binding site, and then it has a dimerization domain where two of these proteins they form a dimer. And so one of the mechanisms for DNA condensation or interaction with DNA, it's this bridging. So it forms some sort of bridging between two DNA molecules. But the action also depends on the concentration of magnesium. So it has been shown that if there's no magnesium, that instead these proteins can form large aggregates, which then, instead of bridging, they stiffen the DNA instead. So one of the problems, of course, then, of studying crowding effects is that, there's a lot of molecules which decreases the number of techniques that we can use. And so most of the work that has been already published on similar topics, some sort of protein and crowding, has been done mainly with analytical ultracentrifugation, followed by gel electrophoresis. And there's also been some work on fluorescence, fluorescence microscopy. So what we've decided to work with was with fluorescence correlation spectroscopy, because in this case, we can actually just tag the DNA, so it's fluorescently tagged, and we can then follow the diffusion. So this is just a plot that shows you then the translational diffusion of the fluorescently tagged DNA. Here is just an example of an autocorrelation function and the corresponding fitting, not going into too many details. What I want to point out is here on the top, on the red curve, we see that then the translational diffusion of the DNA as we increase the concentration of HNS. And what we see here is that there's actually no significant variation of the translational diffusion. So there's no obvious condensation, if you wish, of the DNA. If we look at this axis here, we look at how the translational diffusion of DNA varies with increasing PEG. And we see that there's a decrease. And of course, I'm using here PEG as a crowding agent and is not as inert as one might hope. So of course, there's some hydrophobicity. There's changes in the electric constant and so forth. But anyway, if we do follow just keeping one PEG concentration constant and then just follow how the diffusion of the DNA molecule changes with the protein concentration, we see that, for example, for the higher PEG concentration that we used, which was 6%, we see in this particular case, we see that there's a clear decrease of the translational diffusion, and then is followed by actually an increase. And these points here are actually aggregation. So we see the aggregation of DNA. We can actually see it visually and in the microscope. Okay, and I should also say that this is pretty much consistent with what was observed before using dynamic light scattering. Concentrations of HNS which are not too high, they don't actually have a significant impact on the nucleoid structure. So this was some work done by De Vries in 2016. But this was measured just using dynamic light scattering, and then, of course, there was no measurement with the crowding agents. They used a fluorescence microscopy instead. So what we wanted to do next, so this seems like there's some condensation going on, which is, so there's some, driven by the presence of both the protein and the PEG. And we also wanted to see how does the binding of the HNS then protects DNAs towards the action of enzymes. And we used a couple of different enzymes. So we started off with just using DNase, which basically just chops all the covalent bonds between the nucleotides. So it has no specificity at all. And when I'm talking about specificity, it's just a base pair type of binding specificity. And here, so what we see here is that here is a bound for the free DNA. So we use, I've mentioned we use a long linear DNA molecule. So we're not taking into account supercoiling at all here. And then we see that if we, in the lane 2 here, we have then our free DNA, which is digested by DNase, and we see that it's quite much digested. We just see very small bits that travel long in the gel electrophoresis, in the electric field. And then the addition of HNS, so we see that there's some protection, but not to the same extent. So it's not able to protect all molecule. On the other hand, if we now have the PEG, then we can see that there's actually some protection going on. So we don't have as many small fragments as we did in the absence of the crowding agent. And here's just a control experiment showing that in the presence of PEG, the DNase enzyme is still active. Now, I mentioned that that the HNS has low to medium specificity. And so it binds a little bit, in principle, all over the DNA, but it prefers to bind on the AT regions. And these are regions that are a little bit more flexible. And they are also commonly found close to the promoter region, where the RNA polymerase binds to start the transcription of a gene. So what we've been looking at is actually, we chose 2 specific enzymes. One of them, so if we look here just at this plot, the green lines here, they show the content of GC along the plasmid, the linearized plasmid that we were using. So, and we chose 2 different restriction enzymes. One that binds more specifically to it binds to an AT region and cuts, and one that binds to a CG region and cuts. Okay, so basically, because there's only one binding site for each one of these proteins, every time we have this DNA molecule with these enzymes, it will just chop the DNA in 2. And this is what we can see here, for example, for the NDE1, which binds to the AT-rich sites. We see that we have the free DNA as before, just one bond. And then if we digest it with this enzyme, it will cut this DNA into 2 bits. Now, if we add the HNS to this system and digest it with our enzyme, we see that it's actually above a certain HNS concentration, we have a protection of that particular site. So it stops digesting. On the other hand, if we look at the NRU, which is the enzyme that chops specifically on a GC-rich site, then we see that there's no protection, not even for the larger HNS concentrations that we use. So what happens with the PEG then, if we add our crowding agent to this? What we see is that in the case of the NDU1, which again binds to the chops the AT regions, we see that the protection, so we have exactly the same concentrations of HNS here, but in the presence of PEG, and we see that the protection actually starts at lower HNS concentrations than it did in the system without the crowding agents. On the other hand, when it comes to the GC sites, rich sites, then there's no real significant protection compared to the system with and without crowding agents. So there's been a lot of work, of course, looking at the effect of crowding in a number of biological, in a number of reactions. And and we know that work by Minton and many others that the crowding affects changes in the association rate of aggregation, and it affects binding constants, and it affects the changes in conformational behavior. So it seems that when we have this HNS-PEG system, that both are actually playing a role. So it increases the binding, and it does also induce some change in the conformation. So we wanted then to check if this is specific for a DNA binding system. And this, again, has already been done already by Zimmerman. So he, for example, sort of extracted the cell lysates and then added that to the DNA. And he was also looking at other BSA and PEG and so forth. So there was quite nice work done already in the 90s. But again, using ultracentrifugation. Analytical ultracentrifugation, we wanted to use something else. So we started, and we took 2 different molecules to look at this non-specificity of the binding agent. And first, we did use spermine that we've already saw before. So we know that spermine condenses the DNA. And to look at this, we were using dye exclusion assays. So this is a very simple assays. We just we use a dye that, when it binds to DNA, increases a lot the fluorescence, and then we just add our binding agent, and we see how the dye gets excluded from the DNA, and we can follow that by the changes in the maximum intensity. And this, of course, means that the DNA is that there's some competition between the binding and the spermine in this case. So it doesn't necessarily mean that the DNA is condensed. It could just be well protected. But we know that, for example, from fluorescence microscopy and from electron microscopy, that it does, in fact, condense. So we're just looking at this intensity. And we sort of plotted it like that instead. So this is the same, similar data as we've seen before, just seeing how the intensity, normalized fluorescence intensity to the DNA alone, changes with increasing the spermine concentration. You'll notice that condensation is very weak. And that is because we're using a buffer with a relatively high ionic strength. So we have the interactions are screened. The electrostatic interactions are somewhat screened. But then what we see is that as we increase the amount of PEG, we see that there's this huge decrease in the intensity. So for example, for even at very low concentrations of spermine, we see that there's a reasonably strong condensation when compared with a system without PEG. So this is synergism is also found for non-specific DNA binding agents, and again, this was found before. We wanted to try yet another system, and this is also inspired by the fact that the protein also self-assembles. So we wanted to take a non-specific self-assembling DNA condensing agent or DNA binding agent. So I took a cationic surfactant. As we've heard yesterday, I was working with that system during my PhD. So I feel somewhat comfortable with it. So here's just showing, again, that we have the same type of experiment when we're looking at the normalized fluorescence intensity as a function of the CTAP concentration. And the field circles here, they correspond to the DNA CTAP alone. And we see the decrease. And then in the open circles, we have the PEG as well. So here we use a lower concentration of PEG. And we see again that there's a general decrease. But if we look at the salt effect now, so in this case, we're working with 10 millimolar tris buffer, and we increase here to 10 millimolar sodium bromide. Then we see that this synergism or this joint CTAB and PEG becomes a lot stronger. And this makes sense because we know that we're decreasing, from one hand, we're decreasing the electrostatic interactions between the DNA and the CTAB by increasing salt. And on the other hand, we're facilitating the DNA condensation by the psi condensation, polymer salt-induced condensation. So it's also clear here that, that molecular crowding then lowers the CAC of the CTAB in the presence of DNA, because that's also something that we know, is that the CTAB does not condense DNA if it's just as a unimer. It's not enough. So only when it forms micelles in the presence of, in the vicinity of the DNA, which is the CAC, does it start to condense. So clearly there's a very large effect of the CAC in this case. And we wanted to check this. So our collaborations in BUT then, they were looking at the CMC. So we wanted to see if this decrease in the CAC also was a consequence of a decrease in the CMC. So just the critical micellar concentration of the surfactant in the absence of DNA. And so we were using ultrasonic spectroscopy to do this. And here is just an example of a curve where we can look at the measurement of the CMC. And here's just a measurement with water, so 0.9 millimolar, which is very consistent with the CTAB CMC. And then we see that by having 10 millimolar of buffer, so that was our conditions, it decreases a little bit, as we would expect. It decreases even more if we have the 10 millimolar of sodium bromide. But what was interesting is that actually the presence of PEG did not, or at least for the concentrations of PEG that we used, did not actually affect significantly the CMC. So it seems that, that whatever effect is going on here, so it seems like the PEG is affecting the DNA conformation more than, well, obviously the surfactant micellization. So, so this, I've been talking about PEG, and I just wanted to, so we were interested in looking at other type of crowding agents, and particularly we were really interested in looking at negatively charged crowding agents because, well, most of the macromolecules that you'll find in the cytosol are negatively charged. So of course, a very naive view of the cell, my own, would be that, okay, why don't these proteins just spread around and start interacting with the crowding agents? Why do they keep with the DNA? So we were just like trying out a little bit with that. And so we started off by, for example, looking what happens if we use a negatively charged polymer. So we just used PAA. And we see that there's, so this is the plot with DNA CTAB again. And here is when we add 25 milligrams per milliliter of PAA. It doesn't condense at all. So that is, it wasn't surprising. There's even been some simulations done on this. So PAA prevents DNA condensation by CTAB. But we were interested in knowing what happens with a sort of spherical. So for example, a ribosome. We can think of a ribosome as being a negatively charged sphere. Now, we don't have experimental results on that part yet, but I just wanted to very briefly show you some simulations that we've been doing. So very, very briefly, because I think my time might be running out soon. So this is our extremely simple model. So it's even more coarse-grained than many of the coarse-grained systems that we've been looking at these 2 days, but anyway. So here we have our DNA as just some hard spheres with negative charge and harmonic bonds. We're mimicking our HNS protein, or our DNA-binding agent with a dimer with positive charge in each. And then we have our crowding molecules, which are these bigger spheres, and then we have the counterions for both. And this is a primitive model, so we have the water comes in as a dielectric constant. So what we've done here is 2 things. One, we have increase the self-association between the dimers. So we're inducing self-association, so like a C-tab mimicking C-tab or a protein that associates. And second, we have then increased the charge of these crowding molecules to see what happens. And, well, these are our equations. I'm very happy to go through them afterwards if you're interested. Here, just showing, it's not so interesting, so I think I will pass by. OK. So here we have so probability of the radius of gyration of the polymer, DNA, as well, probability of the radius of gyration. And here in this case, so the different colored curves correspond to different crowding densities. So we're increasing the crowding density. And in all these systems here, the charge of the crowder is 0. So it's neutral crowding agents. And what we do from this plot to that is to just induce a self-association between these dimers. So for example, for 0 kT, then we just have an extended chain. It's not even neutralized. This is the size distribution of a neutral chain of equivalent number of monomers. This is the chain with only the counter ions. We have a one-to-one charge between the proteins and the DNA, which is far too high on a cell context, but nevertheless. And what happens is that when we induce a self-association between the proteins, we compact the DNA. So we see how these distributions become narrower and they move to average shorter sites. And this is the 2 kT. So it's not even a very strong. So we use a Lennard-Jones potential to induce this hydrophobicity. And it's important to note that this 2 kT is not enough to aggregate the protein dimers in the absence of the DNA. So it works exactly as the DNA C-tab system. So we see here how, and this is like our blob that so the condensed state. So what happens now if we start, so we're going to take up this system here with 1 kT, and we're going to increase the charge of our crowding agents. So we start with this system here with with the crowding agents are neutral. And then if we increase the charge density, then we see that it starts shifting into larger average radius of gyration, and the distribution becomes broader. And it goes on like that, and then it just decompacts completely. So there's some clear, and here is just some radial distribution functions showing that, well, the binding agent just spreads equally throughout the cell, and it decompacts the DNA. So very similar to what we see with the PAA. And now, on the other hand, if we use the 2 kT system, then we're able to increase the charge density and still keep it at condensed. So they start condensing a little bit with the crowders. There's definitely some interaction with the crowders, but it keeps the stability. So just to summarize then, so we've been looking at the synergistic role of DNA-binding agents and macromolecular crowding on DNA condensation. And we saw that crowding induces both DNA conformational changes and affects the binding. I didn't go into this, but we believe that the PEG acts on the DNA-HNS complex because it forms loops, as we can see here in this AFM image. We saw that synergism is not specific to DNA-binding proteins. We saw that there's a big effect also on the ionic strength. And finally, we suggest that protein self-association leads to a stronger DNA condensation and also to a more stable protein DNA complex. And well, that's all. Thank you for your attention.
Questions?
Shall I start? Okay. It was interesting, this effect on PEG on CMC of C-tab.
Yeah, I should have talked to you before.
Yes. What had you expected?
I actually actually thought it would go down, that the CMC would decrease, because it has been, like for example, there has been some work done with that it decreases the critical concentration of actin fibers, for example, inside the cell. So that has been shown, but you don't agree?
Almost anything polar, non-ionic that you add to water increases the CMC.
Yeah.
So it may be two compensating effects.
It might be.
If you add polyethylene glycol, CMC increases.
Okay. So. But then I suppose one needs to go to higher concentrations?
Maybe, yes, yes. But
I had two questions actually. One question is about you show how much the PEG had. I mean, you showed extreme effects of that, and you also studied the concentration. What about the molecular weight?
Yes, no, we didn't look at the molecular weight. That's true. We bought it, but it never happened.
It can have a effect there.
Yes, definitely, and it does in many.
Yeah. And my other question was that you used that was polyacrylic acid.
Yes.
How would it be, you know, with this new way to do synthesis? How would it be if you had a PEG, for instance, and then you could have different lengths of negatively charged blocks? Would that be of interest for you?
That could be interesting. Yes. Yes.
Yeah, just to follow up Bjorn's comment.
Yep.
Bernard Caban did a lot of work on this, and you should look it up.
Yep.
He worked more with SDS, but I think the effect would be about the same.
Yes.
Last question.
Another question. I'm curious to know how do you perform the experiment with the PA? If you had, I mean, the three component differently, would you get the same situation or morphology at the end?
Could you repeat it?
How do you had the different components?
You mean the mixing order? Yes,
DNA,
yes. No, that's a very good point. Yeah. So we initiate. So we typically start with the DNA, and then we add the binding agent, and then we add the crowding agent in the end, and we leave it to equilibrate so that it's a little bit faster. So we hope that that equilibration is a little bit quicker than, but certainly mixing. But certainly, if we will change the mixing order, it will probably be a little bit different. I mean, the overall behavior should be the same, but the borders are probably going to change, like when it starts compacting and things like that.
Okay, let us thank the speaker once again.